Welcome to Professor SNAB, where we aim to make A-level biology as simple as a cell. So today we're understanding how isolation reduces gene flow between populations, leading to allopatric or sympatric speciation. It's the development of new species from pre-existing species over time. Two populations of a species must be isolated for speciation to take place. The exchange of genes can no longer take place, and this gives rise to two species. There are two types of isolation that can lead to two different types of speciation, and one is geographical isolation, which results in a speciation type called allopatric speciation, and random mutations that prevent interbreeding, which leads to sympatric speciation. Takes place because the isolated populations have different selection pressures in the environment, and different alleles will be prioritized in different environments. A process called genetic drift can take place, and this is when random mutations in alleles take place. And over time, the two species can no longer interbreed to produce a fertile offspring. So it is at this point we can confirm that speciation has taken place. So what is allopatric speciation? It is the result of geographical isolation, and this arises as a result of geographical barriers which can be both man-made or natural. So this can be a river splitting down a land, so then the species are split into two geographically, or it could be a man-made thing like a dam being built in the middle of a river. So that leads to two different types of beavers, for example. Just a random example, it's not something for you to remember, but yeah, so that's one way geographical barriers can lead to allopatric speciation. Changing alleles frequencies result in changes in the phenotype of the two populations. What is sympatric speciation? This is a type of speciation that takes place without a geographical barrier. Isolation instead occurs when random changes in the alleles and therefore phenotypes of some individuals in a population prevent them from successfully breeding with other individuals in the population. Examples of phenotype changes that lead to isolation include possibility of developing different mating or flower seasons, possibility of changes in genitalia, possibility of changes in courtship behaviors, and as the difference in allele frequency increases, the isolated species will no longer be able to interbreed to produce fertile offspring, as they no longer have gene flow and two separate species form. So that is all from Professor SNAB today. Hope you found this topic useful. If you have any questions, let me know in the comments below, and I will get back to them as soon as possible. And don't forget to like, share, and subscribe.